Okay, so for the vesicle formation, you see that they need the support from the coat protein, and there are seven types of coating protein, as I already introduced to you. So for the glycerin coat protein is the best study protein coat protein for among the other coat protein, and also the best study vesicle is the glycerin coated vesicle. So the glycerin coat vesicle, but from both the Golgi apparatus on the outward secretory pathway and from the plasma membrane on the inward endocytic pathway, as I already showed you. So as the plasma membrane, for example, each vesicle will start uh, will start up as glycine-coated pit, and glycine molecules assemble into a basket-like network on the cytosolic surface of the membrane. And it is this assembly process that starts shaping the membrane into vesicle, and how the glycine protein can help to form the vesicle. So when you look at this structure of the glycine, one glycine molecule, that means one glycine protein. So when the scientists they try to isolate the glycine protein from the cell, and to see how this protein behave, uh, and then they see that in a test tube, the glycine molecule sometimes they will cell assemble into the basket-like cage. Uh, Here is a basket-like cage. So the structure of one glycine cage was determined by the cryo-electron microscopy, and in this figure is one cryo-electron image, microscopic image. You can see the basket-like cage of the glycine vesicle. It is one like glycine vesicle. So each glycine, they have the six polypeptide chain, and three of them are heavy chains, and the other three are light chains, and they arrange as a three scalene structure as you see here. So each glycine protein assemble as they stay as a three scalene structure. So that is a functional structure of the glycine protein. And on the heavy chains, on each heavy chains, the head of the heavy chains, they are the binding site for the assembly particle. That means the other glycine protein, they can come and interact with the other glycine protein through the binding to as the heavy chain, as the head of the heavy chains here. So by the assemble of the glycine molecule, so they can make or form a basket like case, and that is the shape of the vesicle. You can see the step of the each step of the vesicle formation process. So this is a electron micrograph showing the sequence of events in the formation of a glycine coated vesicle from the glycine coated pit. So whenever the glycine bound to the membrane, as I said before, so as the plasma membrane, each vesicle start up as structure they call the glycine coated pit. Okay. So the formation of the coated glycine coated vesicle from glycine coated pit is shown in this electron microscopic image. Um, in this image, also the clathrin-coated piece and the vesicle are unusually large and are being formed at the plasma membrane of a hand oxide. Hand uh, oxide to okay, and they are involved in taking up the particle made of lipid and protein into the oxide to form the yolk, a structure inside the oxide for storing the nutrient to the development of the embryo as the early stage. So you see that the image showing the numbers of clathrin-coated pit and vesicle budding from the inner surface of the plasma membrane. Sorry, in this the other image. Okay, so you see at the beginning when the clathrin concentrated at the inner part of the plasma membrane, and then the plasma membrane is blended, um, and after that the budding is formed and developed, and then a vesicle is formed, and this vesicle will be pinched off from the plasma membrane by the dynamic, and then this vesicle will be transported inside the cell to the other organelle like endosome or lysosome. So this is one image illustrated about the vesicular form from clathrin molecule. You can see the video clip for this formation. I hope it works.
Eukaryotic cells take in extracellular molecules through.
Sorry, I think I have to start again. This. So wait me a little. Okay, so this video clip illustrated about the formation of clathrin-coated vesicle.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles. This movie shows a series of electron micrographs that have been artificially morphed to show the process of endocytosis as it may occur. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions. During the assembly of a clathrin coat, the individual triskelions come together, interact through their leg domains, and ultimately form a closed cage.
Okay, so I think it's quite interesting movie about the vesicular formation. and also the scientists who discovered the glycogen protein together with the other protein involving in the vesicular transport, they get the Nobel Prize, as I told you last week. Okay, so I will back to the PowerPoint. Okay, so I think through the video clip you already maybe you understand a little more about how the vesicle is formed and the function of the protein, like adaptin protein or coat protein or dynamine protein during the vesicle formation. So the next, after the vesicle is formed, and the next step is about the translocation of the vesicle on the in the cytoplasm. And as I said, that this part you will learn in the cytoskeleton part teach by Dr. Long. So I will quit this step and we move to the third step of the vesicle transport, involving in the vesicle tethering. So for the vesicle tethering, they need the support from the tether protein. So as I say that after the vesicle is formed and translocated close to the target compartment, so before they do the fusion with the target compartment, they need to confirm that the target compartment is the right target located for this vesicle, and to check the information, so they will use some type of the protein expressed on the vesicle membrane. They call the tether protein. This tether protein, they also say on this, they are complex of the protein, and they express both on the vesicle membrane and also on the target membrane. So it's somehow like when you imagine for the boat, this boat, they come from this country to the other country. For example, they transport the cargo from China to Vietnam, for example, like that. So they need to come to the like Haiphong port. Okay, so before the boat come into the port, they need to do the tethering step, and they stop at a distance from the port, and then they will make a call to the port, something like this, to check the information that is this the Haiphong port, and I have the cargo with this name, like something, and then they need to be confirmed together. So if it's not the Haiphong port, for example, they have to turn back and go to the other way. So for the protein vesicular transport, it also do the same way, that the vesicle come close to the target compartment and they stop, and then they do the tethering to check the information between the vesicle and the target compartment. So this checking somehow is like the interaction between the protein on the vesicle membrane and the protein on the target membrane. So the function of the tether protein is to promote the specific localization of transport for vesicle on target compartment membrane, and they act prior to the formation of the SNAC complex, another type of the protein responsible for the targeting and the fusion of the vesicle on the target membrane. So just for a moment. So for the theta protein, uh, for now the scientists identify the two pro classes of the theta protein, and they can be either long coil protein. Here, the long coil protein. That means protein, the polypeptide chain, they coil together. Okay, or they can be like multi-subunit tethering complexes. For example, here, this is the multi-subunit tethering complexes. They contain many like Cô xin lỗi cả lớp bởi vì mạng nhà cô lại vừa mới đẩy out mất. Hôm nay mạng của nhà cô rất là trục trặc từ sáng đến giờ nên là chúng ta bị mất nhiều thời gian quá vì việc này. Okay, so I hope from this time point it will be fixed and as the internet is fixed, and I hope that we will not miss this problem again. Okay, so for the tethering step, so they need support from tether protein and the energy from the GTPA hydrolysis. So on the vesicle membrane, you can see there are some protein, and these proteins they can interact specifically with the protein on the target membrane. So after the interaction, so the cell can the vesicle will be come closer to the target membrane to do the fusion. Okay, so we will see how this process can happen. Okay, so in this slide they illustrated the step of the docking or after the shattering and the docking of the vesicle to the target membrane. So on the vesicle membrane, there is RAP protein or RAP GTPase illustrated as a yellow color particle attached on the membrane of the vesicle here. So that is the RAP protein, and RAP protein also belongs to the shattering protein. So this RAP protein is recognized by a specific protein on the target membrane. This the green fiber protein, and green fiber protein also belong to the tethering protein. So each green tethering protein here will recognize a specific RAP protein spread on the vesicle transport membrane. So only when this interaction happens between the RAP and the tethering protein on the target membrane, so the vesicle will be bring close to the membrane of the target compartment. So this process is called the docking. The docking. That means they bring the vesicle close to the target compartment. And after that, when the vesicle is close enough to the target compartment, they will do the fusion. And the fusion happen depend on the work of the other protein they call the snare protein. So the RAP and tethering protein provides the initial recognition between a vesicle and its target membrane. And in the next step, for the fusion step, the snare protein also do the recognition of the vesicle and the target membrane. And the snare protein will ensure that the transport vesicle dock at the appropriate target membrane. So you see, the cell does a double check for the vesicle and the target localization or the target destination. So they check the first the first check was is the interaction between the tethering protein on the vesicle and the tethering protein on the membrane, target membrane. And the second check is between the snare protein on vesicle and on the target membrane. So that double check for the specific um, destination of the vesicular transport. So for the docking and fusion step, they need the support from SNAP protein. The SNAP protein, in fact, this is one family of the protein, and the most SNAP reside predominantly in specific cellular compartment. That means, depend on the vesicle, they will also have the different SNAP protein, and because of that, the SNAP protein will reside predominantly in specific cellular compartment. And these SNAP proteins, they regulate the vesicle docking and fusion state, the last state of the vesicular transport. This family, SNAP family, they are small tail anchor protein. So they are like the tail anchor protein, and the size of the protein is quite small. Mm, thus, in every SNAP protein, they contain the SNAP motif as expressed as a blue part, blue color part in the image here. And this motif, they are very conserved. That means they share this common amino acid sequence between every snare in every species. And in this motif, they contain around 60 to 70 amino acids and form the coil structure. The two snares, when they interact together, they will form the coil structure as the snare motif here. The snare motif of the V snare, that means the snare on vesicle. So you see in some image, they write like V snare, that means the snare on vesicle, and T snare, that means the snare protein on target compartment. So all of them, they belong to the snare family, but depend on where they located on the vesicle or the target. So they can be the either V snare or T snare. And when the V snare and T snare they interact together, they assemble to form the trans snare complex. And specific pairing between the V snare and T snare is the key step in the membrane fusion. And also, specific pairing means the double check between the vesicle and the target membrane. So this is a snare complex, trans snare complex made from the V snare and the T snare. This is one example of the docking fusion on the vesicle and the target membrane in the neuron. So in the neuron cell, the V snare on the vesicle, it is synaptobrevin, synaptobrevin. So the name of the snare, the V snare on the vesicle, and on the nerve cell plasma membrane, there is located a localization of the T snare in syntacin. So syntacin and synaptobrevin, they recognize each other, and then they do the coil complexes to make the trans snare complex. They call the this is a trans snare complex. Also, you see they also need the support from the other T snare called the SNAP 25. So together, three types of the snare make a complex, and when they make the coil, they will uh, they will recruit the vesicle come closer to the target membrane. Okay, I will quit this part. So following the docking, that means the bringing of vesicle close to the membrane. The SNAP protein can catalyze the fusion of the vesicle and the target membrane. So when the vesicle come close enough to the membrane, so the V-snare on the vesicle will make the coil with the will interact with the T-snare to make the trans-snare complex. Here, you see. After that, or when the this pairing is triggered, so the ties pairing of V-snare and the T-snare draw the two lipid bilayer into a close opposition. So somehow you understand like this like the two zipper in the two trans snare complex. So when the zipper is closed, they will make the vesicle membrane come close to the target membrane. The force of the snare winding together squeezes out any water molecule that remain trapped between the two membranes, allowing their lipid to flow together to form the continuous bilayer. So, in a step, other protein recruits the fusion site help to complete the fusion process. After the fusion, the snare are pried apart so they can be reused again. So in the next slide, I will see that. But here, the key point in this step is that the V snare recognize the T snare together and they start interact, and the tie pairing between the V snare and the T snare will draw the two lipid bilayer of the vesicular membrane and the target membrane into close opposition, and the force of the snare winding between the two winding together will squeeze out, that means remove any water molecule that remain trapped between the two lipid bilayer of the two membrane here. And this water removing will allow the lipid molecule from both membrane to flow together to form a continuous bilayer. And after the fuse of two membrane, so you can see all the cargo in the vesicle will be on the target compartment or on the membrane of the target compartment. So after the fusion of the membrane of the vesicle and the target, so this trans snare complex will be a dissociation in order to reuse the snare on the membrane. So the disassemble of snare complex can have happen with the support from the other complex they call the NSF and adapter protein. So NSF and adapter protein, they can bind to the trans snare complex and use the energy from the ATP hydrolysis to separate the two snare in the trans snare complex. So after that, the V snare and T snare can be separated and stay on the membrane, and they can be used to another cycle of the vesicular transport. So one ATP is used in this step to disassemble of the snare complex. For the GTP hydrolysis, uh, in this slide, this schema shows about how the RAP protein help to ensure the specificity of vesicle docking. So in the tethering step and the docking fusion step, you see during the first step of the vesicular transport, when the vesicula is formed, on the membrane of the vesicula already localizing up the RAP GTP. So the RAP GTP is spread as a yellow circle with the GTP molecule here, and also the Visne. Okay, so then the vesicle is pinched up and transported on the cytoplasm to the target compartment. So when they are close to the target compartment, they do the chattering process by interact of the RAP GTP with the RAP effector on the target compartment. So remind that you should note that the RAP GTP and RAP effector here, both they are belong to the tethering protein. So when the RAP effector they bind to the RAP GTP, so the binding of the RAP effector and the GTP will trigger the docking of the vesicle to the target membrane. And when the vesicle bring close to the target membrane, the Visne and the Tisne interact together and makes the trans-SNE complex. And then the vesicular membrane is fused with the membrane of the target compartment. After the fusion, you see the RAP GTP and the RAP effector when dissociated, and the RAP GTP on the membrane also released from the membrane of the target compartment by the GTP hydrolysis. So after GTP hydrolysis, now the RAP will bind to the GDP and becomes the solute RAP GDP. The solute RAP GDP, the solute RAP GDP is bind with the GDP dissociation inhibitor called GDI. So the green, that is the GDI, will help to maintain the binding of the RAP GDP, the solute RAP GDP, and together this complex will be bring back to the donor compartment and interact with the GAP protein on the donor compartment. So the GAP here means guanine nucleotide exchange factor. So now you see this similar to the transport, the run GIP and the run GAP on the membrane of the nucleus, right? So this GIP will exchange the GDP by the GTP and RAP now by to GTP and ready for another cycle of the vesicular transport. Okay, so that is one cycle of one transport on vesicular transport from the donor compartment to the target compartment, and they use one GTP to do this one cycle. And the GTP hydrolysis involving in the step when the RAP GTP is bring back to the donor compartment. So for the vesicular transport, it is energy requiring, and the energy is used as the first step, the vesicular formation, and the fusion docking, and then the reuse or the bring back of RAP GTP protein to the donor compartment. Okay, I will increase this. So the transport between the ER and the Golgi apparatus also belong to the vesicular transport. And The transport between these two organelles, they are two direction. That means they can either the biosynthesis secretory pathway from an ER to the Golgi or the retrievable pathway from the Golgi to the endoplasmic reticulum. Okay, so when we look closer to the formation of vesicle from the ER to the Golgi apparatus, in previous slide, you see the formation of Clathrin coated vesicle, and now it is coated vesicle. So as I said to you before, that the vesicle is formed from the ER by the support of the coat protein named the COP2. So COP2 makes the vesicle on the ER. Okay, remember that. So in this case, the cargo protein, like the solo cargo protein in the ER lumen, they need to bind to their specific receptor. So this is the cargo receptor, the green one. So whenever the solo cargo protein bind to the cargo receptor, they will change the outer part of the receptor, and as a new conformation is expressed like the exit signal on the cargo receptor. And this exit signal will be recognized by the on-type adapter protein named SAR1, SAR1 GTP. So SAR1 is the adapter protein for the COP2 on the ER membrane. So SAR1, when they will interact with the receptor and bind to the membrane. So the presence of the SAR1 GTP will recruit the other co-protein like the red and the brown, the red and the orange protein here. They belong to the COP2 protein. They are the subunits of COP2 coat protein. So the SAR1 bind to membrane and recruit the COP2 protein to come and form the coat on this side of the membrane to make the vesicle. So after that, this vesicle is transported to the Golgi apparatus. So here I would like to help you to understand more about how the adapter protein they can interact with the membrane. So at the in the A, that is a structure of the SAR1 protein. So SAR1 also belongs to the small GTPase family. So the is a schematic of the schematic diagram of the SAR1. And you can see the COMSTOF domain are predicted on the structure of the polypeptide chain. The N terminal stay as amphipathic alpha helix, and they have two switch regions, switch 1 and switch 2, and the interswitch region is spread as the purple color here. So this is the structure of the SAR1 protein. In the B image, showing the shark protein cycle between the membrane association and dissociation. So at the beginning, when the SAR1 stay in the cytosol, so they stay as a GDP bound cytosolic SAR1 protein, and they are is inactivated, and they carry the N-terminal amphipathic helix in the hydrophobic pocket. So the the alpha helix, N-terminal alpha helix is hiding in the hydrophobic pocket. So when the exchange, the GIP here, that means valine nucleotide exchange factor. Okay, again you see the GIP exchange the GDP by the GTP in the SAT1. So the GTP loaded SAT1 undergoes a conformation change of the two switch here and the interswitch region. So both three domain will change will change the conformation and they trigger the extrusion of the helix from the pocket. So now the amphipathic alpha helix as the N terminus is extruded and spread. Um, Subsequently, the shallow insertion the amphipathic helix into the outer lipid of the lipid bilayer and allow the SAT1 to associate tightly with the membrane surface. So at this moment, the SAT1 will bind to the membrane of the vesicle here. And for the dissociation, the GTPA will activate the the GAP will activate the hydrolysis of the GTP, and then the SAT1 will bind to GDP, and they will come back to the first conformation, the converse conformation, with the hiding of the amphipathic alpha helix as the N-terminus, and they cannot bind to the membrane. Okay, so that is one cycle of the membrane association and this association of the adapter protein named the SAT1 protein. So this the work of the GIP on the SAT1 protein is triggered by the binding of the cargo onto their receptor on the membrane, and they will trigger the SAT1 GTP come and bind to the membrane of the vesicle. So, so I just want to illustrating for you to understand more about how the adapter protein can come and bind to the membrane. So for the COP1, they need support from the SAT1, and for the COP2, for the COP2, they need the support from the SAT1, and for the COP1 complex on the Golgi apparatus, the adapter protein now is the RF1, the ARF1 GTP. And also the behavior is similar to the SAT1 GTP. Okay, so I will quit this slide. Okay, so the COP2 coat makes the vesicle from the ER, and then this COP2 coated vesicle is transported to the transverse of the Golgi apparatus and fused with the Golgi apparatus to release the cargo to the Golgi lumen. Also from the ER and the Golgi apparatus, there is the retrieval pathway, as I already said to you. So for the retrieval pathway, it is a way that the cell will reuse some protein expressed on the membrane of the ER. So these proteins, they can be transported maybe randomly to the Golgi apparatus, and then they need to be sent back to the ER membrane. So for the here is one process illustrated about the retrieval pathway. Okay, so note that all the proteins stay in the ER. Oh, sorry, it is 10:55. So we have a short break. I'm sorry, I didn't see them.